Good afternoon to those of you in the United States, and good evening to those of you logging in from Europe. Welcome to this webcast presented by Clinical Lab Products Magazine, your source for the latest about tools and trends affecting the clinical laboratory community. The topic and title of today's webcast is Detecting Pi3k Signaling Pathway Mutations. My name is Steve Hallisey, Chief Editor of CLP, and it will be my pleasure to moderate today's event. Before we begin, here are a few housekeeping items about today's program. At the bottom of your console or in the chat box, you'll see that there are a number of commands that you can use. We will have a short Q&A period at the end of the presentation, but we encourage you to submit your questions at any time during the webcast by navigating to the chat box and clicking on the questions arrow. We'll try to answer all of your questions during the Q&A period, but if a fuller answer is needed or if we run out of time, they'll be answered later via email. Additional materials for today's webcast, including a list of the numbered references cited in the slides, are available via the handouts arrow. If you're viewing the on-demand version of this webcast, you'll note that a PDF version of the slide deck is also available by way of the handouts arrow. The on-demand version of this webcast will be available in just a couple of days after the event, and it can be accessed using the same audience link that was sent to you earlier. In a moment, I'll introduce the speaker for today's webcast, but first I want to acknowledge the company that is sponsoring this event. With U.S. headquarters in Germantown, Maryland, and international headquarters in Hilden, Germany, QIAGEN is a multinational provider of tests and instruments for the clinical research and diagnostic community. The company's diagnostic systems are used in complex biomedical testing and are found in hospitals, reference laboratories, and physician office settings around the globe. So now to the topic of today's webcast, Pi3k testing. Patients with advanced breast cancer often develop resistance to endocrine therapy. Studies suggest that resistance to therapy and disease progression may result from activation of the Pi3k pathway, known to be the most frequently altered pathway in human cancers. The PIK3CA is an oncogene that codes for the Pi3k alpha isoform and may be mutated in up to 40% of patients who present with advanced breast cancer. To aid in identifying breast cancer patients with a PIK3CA mutation who may be eligible for treatment with alpelisib, a novel alpha-specific Pi3k inhibitor, a companion diagnostic for PIK3CA is needed. Joining us today to help us understand the methods for finding Pi3k signaling pathway mutations is Lawrence M. Weiss, MD, who is Chief Medical Officer at Neogenomics, a high-complexity CLIA-certified clinical laboratory, pharma services, and information services company specializing in cancer genetics diagnostics testing. Before joining Neogenomics, Dr. Weiss was president of the medical staff and chairman of pathology at the City of Hope. So I'd like to get out of the way now and let Dr. Weiss take over. Dr. Weiss, go ahead.
Thank you very much. As stated, we'll be talking about detecting Pi3k signaling pathway mutations, specifically mutations in PIK3CA. Next slide, please. So here are the topics I'm going to discuss. We'll provide a brief overview of hormone receptor positive, HER2 negative, advanced breast cancer, and its resistance to endocrine therapy. We'll talk about the biology and cancer activation of the Pi3k pathway, and we'll specifically talk about testing for PIK3CA mutations. We'll talk about how you have to use a companion diagnostic to qualify patients for treatment. And then we'll talk about the companion diagnostic itself, PIK3CA CDx Mutation Analysis by PCR, which is mentioned as an FDA-approved companion diagnostic. Next. First, the overview of hormone receptor positive, HER2 negative breast cancer. And let's level set on breast cancer itself. Breast cancer has the highest incidence of all the major cancers in the human body, about 266,000 cases occurring in 2018. Fortunately, if breast cancer is diagnosed at low stage, its survival rate is very high. Unfortunately, the 5-year survival rate is about 25% in patients with advanced cases of breast cancer, recurrent or metastatic. Next. Let's divvy up breast cancer by HER2 and hormone responsiveness. And when I talk about hormone responsiveness through this entire talk, I'm talking about not only ER positive, estrogen receptor positive, but any tumor that's either estrogen or progesterone positive. So breast cancers that are hormone receptor positive and HER2 negative make up the largest proportion of breast cancer, almost 75% of cases. The next largest category are the triple negative ones, negative for hormone receptor or HER2 at about 30,000 cases. About 15% of cases are HER2 positive. Next. We can go on to the next slide, please. So, of those 190,000 or 75% of breast cancers that are hormone receptor positive and HER2 negative, about 20 to 30% will progress to advanced stage or metastatic. Of those, 40% are going to be positive for PIK3CA mutation. So that represents about 15 to 25,000 cases. It's a large segment of patients, and up till now, there's not really been a therapy associated with this mutation. Next. Advanced breast cancer does remain a challenge. Endocrine therapy is the current standard treatment option for hormone receptor positive, HER2 negative breast cancer. But resistance to endocrine therapy can occur and will be a challenge when breast cancer recurs or metastasizes. The Pi3k pathway downregulates the expression of ER and PR, in turn reducing estrogen dependence. And the Pi3k pathway plays a role in disease progression and resistance to therapy with advanced breast cancer. Next. So what about the biology of the PIK3CA pathway? Next slide. This is about the most complicated slide I'm going to show. But you can see from the diagram that Pi3k is at the head of a number of pathways that are very important to the cell. It's a key regulator of normal growth processes, including cell growth and proliferation, metabolism, motility through cytoskeleton, survival, and apoptosis. So it's really a key regulator of the cell. Next. And PIK3CA is an oncogene that codes for the PI3K alpha isoform. So let's talk about the PI3K molecules. Generally, they have a regulatory and a catalytic subunit. And we're going to concentrate for the purposes of this discussion. Actually, there's several classes of PI3K molecules. We're just 4 classes, and we're just going to be talking about the class 1 molecules. And we're going to really concentrate on the catalytic subunit. So there are 4 main different types of catalytic subunits called alpha, beta, gamma, and delta. And you can see on their right, their expression varies Some are ubiquitous, some are limited to white blood cells. The Pi3k alpha is ubiquitous. It's encoded by the PIK3CA gene. And that gene is very important in cancer because that is a gene that is mutated, whereas the other genes coding for the other molecules are not very commonly mutated. Next. And alpelisib is an orally administrative alpha-specific Pi3k inhibitor and is only active in cases which harbor PIK3CA mutations. Next. So let's talk specifically about PIK3CA mutation in hormone receptor positive, HER2 negative breast cancer. Next. So actually, PIK3CA is the most common mutated gene in hormone receptor breast cancers. Overall, TP53 mutations are the most common mutation in breast cancers, but they're more commonly seen in basal-like and hormone receptor negative HER2 positive tumors. PIK3CA mutation rates are higher in luminal tumors, which by definition are hormone receptor positive. And it's said that about 40% of patients with hormone receptor positive breast cancer have a PIK3CA mutation. In our hands, since we've started using the companion diagnostic, that figure has been about 38%. So that 40% number in the literature is pretty darn accurate. Next. Now, PIK3CA expression and/or mutation is a poor prognostic marker. First of all, it's associated with a lot of adverse clinical pathologic parameters, and that's what you're seeing here. Actually, this study was performed with PIK3CA expression by immunohistochemistry. And overall, the probability of distant recurrence is significantly increased in cases with increased PIK3CA expression. And therefore, PIK3CA mutations are key disease drivers and are associated with a poor prognosis with advanced breast cancer. That's known for a long time that it's been a poor prognostic marker. But why we're talking about it today is because of its predictive value. Next. It's very important to know your patient's PIK3CA mutation status. As we've discussed, the role of PIK3k signaling is critical to cancer progression, particularly the Pi3k alpha, and many of your advanced breast patient cancer will have a PIK3CA mutation. So as we said, it's in terms of the pathway, it's frequently aberrantly activated and associated with endocrine resistance and disease progression and breast cancer. In particularly, it's the alpha isoform that's important. It's the PIK3CA gene that is commonly mutated, and we know that PIK3k alpha supports tumor angiogenesis and stromal interactions. And so this is the isoform that's important. And as I said, PIK3CA codes for Pi3k alpha, and in fact, it's the most frequently reported molecular alteration of the PI3k pathway in breast cancer. Next. So how does one test for PIK3CA mutation? Well, how it should be tested is through PIK3CA CDx Mutation Analysis. It is a FDA-approved companion diagnostic. The test is intended to aid clinicians in identifying breast cancer patients with a PIK3CA mutation who may be eligible for treatment with alpelisib. Alpelisib is a PIK3CA inhibitor indicated for patients with hormone receptor positive, HER2 negative, advanced or metastatic breast cancer with a PIK3CA mutation. We've heard these are about 40% of cases, and it will progress on endocrine therapy. Next. Just to go over the definitions of companion diagnostics and laboratory developed tests according to the FDA. Companion diagnostic is a medical device which provides information that, according to the FDA, is essential for the safe and effective use of a corresponding drug or biological product. Technically, evaluation by a companion diagnostic is required to prescribe the drug, even though the FDA does not regulate the practice of medicine. In contrast, a laboratory-developed test is a laboratory designed, manufactured, and used within a single laboratory. Of course, LDTs need to be validated as per CLIA rules for individual laboratories. An LDT does not need FDA approval. Next. NCCN guidelines for breast cancer diagnostic testing have been updated to recently added PIK3CA mutation testing that occurred about 6 months ago, and here are the recommendations. So to assess for PIK3CA mutation as part of the initial metastatic breast cancer workup, if all the following is true: the patient has recurrent or metastatic breast cancer, if that tumor is hormone receptor positive, and remember that's either ER or PR, and HER2 negative. So ER negative, progesterone receptor positive, and HER2 negative will still qualify. And one is considering therapy for alpelisib. Next. So what is the eligibility for the companion diagnostic? The criteria for testing is hormone receptor positive, HER2 negative. And when I say HER2 negative, that's either by immunohistochemistry or FISH. Doesn't have to be by both technologies. Patient has advanced, recurrent, or metastatic breast cancer, must have progressed on endocrine therapy, and the specimen is formalin-fixed, paraffin-embedded breast tissue. Could be the primary, it could be a metastasis. The turnaround time is about 7 calendar days. We need formalin fixation. That's the FF and FFPE. Zinc fixatives will not do. And unfortunately, decalcified specimens will also not do. But soft tissue metastases will do fine for testing. Next. In summary, Pi3k pathway activation is highly implicated in cancer progression and estrogen therapy resistance in breast cancer. Mutation of PIK3CA, an oncogene with codes for the very important Pi3k alpha molecule, is the most frequently reported molecular alteration of the PIK3k pathway in breast cancer. In fact, 40% of patients with hormone receptor HER2 negative breast cancer have a PIK3CA mutation. These mutations have been associated with endocrine resistance and disease progression. And expression in advanced breast cancer may be a biomarker of poor prognosis and is certainly associated with other markers of aggressive disease. And specifically, the PIK3CA companion diagnostic mutation analysis, QIAGEN, is an FDA-approved companion diagnostic intended to aid clinicians in identifying breast cancer patients with a PIK3CA mutation who may be eligible for treatment by alpelisib. Next.
Well, thanks, Dr. Weiss, for that great opening presentation. We want to interrupt for just a moment and do a quick poll of our audience and see what their experience has been with PIK3CA mutation testing. So we'll offer a poll here to our attendees, and we ask the attendees to pick the responses that apply there. Dr. Weiss, you mentioned the difference between, while the readers are responding here, you mentioned the difference between an LDT and a companion diagnostic. In your experience, are there a lot of LDTs in this field as well as the the QIAGEN companion diagnostic?
Well, there have been LDTs offered even before the companion diagnostic was available. In our lab, we offered PIK3CA testing as part of a multi-gene NGS panel. We also offered it as a single-gene Sanger sequencing. So there have been a lot of LDTs around for a long time. But now there's a companion diagnostic, and that's the test that one should use to qualify patients for the alpelisib therapy.
Mm-hmm. Well, let's see what our attendees are telling us about what they've done. Okay, there are the results. So more than 2/3 have not ordered or performed PIK3CA mutation testing. Do these numbers surprise you in any way, Dr. Wise?
No, they don't surprise me that much. The drug just became, and the companion diagnostic just became available, I believe, at the end of May this year. So we're still about 6 months out. I think people are just learning about the importance about the drug alpelisib in breast cancer. And I think what we generally see is a small bump at the initially. Then we see a steady rise, and that steady rise occurs over the next couple of years. So we're still early in the game, and but I do think it's very important that patients do get tested.
Mm-hmm, mm-hmm. Well, we'll hide these results and go on to our the next section of the discussion.
So why did the FDA approve the drug, and why is there a companion why is it so important to have a specific companion diagnostic for that drug? And that's due to the SOLAR-1 trial, and we're going to spend a couple slides to talk about that trial. So next slide, please. So what the drug company did is they took a large group of men or postmenopausal women who had hormone receptor positive, HER2 negative, advanced breast cancer. They looked at almost 600 cases, and the qualifications for the study is had to have recurred or progressed on or after or on or prior aromatase inhibitor therapy. They had to have a specimen available to identify the PIK3CA status. They had to have measurable disease, and they had to have relatively good performance status. They did PIK3CA testing on all patients, and they separated them to PIK3CA mutant positive and mutant negative categories. And for each of those categories, they split them into, randomly of course, into groups that got fulvestrant or fulvestrant and alpelisib. So each, the mutant cohort was split into those two groups, and the non-mutant cohort was split into those two groups. They had one important primary endpoint, and that was progression-free survival in the PIK3CA mutant cohort as compared to the placebo group. Secondary endpoints looked at those without the mutation and other various combinations. And of course, safety was investigated as well. And I'll show the results on the next slide. They were impressive as therapeutic results can be. First of all, only patients whose tumors harbor actionable PIK3CA mutations were likely to experience clinically meaningful results. So the survival curve differences in the PIK3CA mutation negative group were not significant. But in the PIK3CA mutation positive group, the median progression-free survival increased from just under 6 months to 11 months. And you can see that in the two curves. And in fact, the risk of progression or death was reduced by 35%. And on this basis, the FDA approved the drug, and they also approved the companion diagnostic that was linked to this study as well. And that's what we're going to describe next. So let's talk in more detail about the PIK3CA companion diagnostic of QIAGEN. Next. So, as stated, alpelisib requires a companion diagnostic. And obviously, before prescribing the drug, the PIK3CA status has to be determined. So the QIAGEN TheraScreen PIK3CA RGQ PCR kit was the first FDA-approved IVD companion diagnostic for this indication, and supports the drug prescription. Next. It's a RGQ PCR kit. It involves, it's a qualitative assay. It uses TaqMan probes and PCR clamp technology that enhances mutation detection. So, as we've been talking about, it's for the identification of breast cancer patients potentially eligible for treatment with alpelisib, and it's a qualitative real-time PCR assay. Obviously includes a control. It's relatively sensitive. The level of detection depends on the genes identified, but the mean mutation allele frequency is about 5%. The sample input is very practical, about 20 millimeters squared of formalin-fixed paraffin-embedded tissue. If you think about that, if you have a resection, it's about 5 by 4 millimeters of tissue. If you have a mammatome biopsy, it's 2 or 3 millimeters by 7 to 10 millimeters. If you have a skinny core needle biopsy, that's about 2 cores, 1 centimeters each. It's also built for a plasma detection as well, although it's not yet validated in our lab, and I'm not aware that it's specifically validated in other labs as well. And material is cut at 4 to 5 micron thickness, which is typical for laboratories. It's a qualitative result, so it's either mutation detected or no mutation detected for each of the actionable PIK3CA mutations, and we'll talk about the specific mutations in a second. And it's very practical, and you can run from 1 to 6 individual samples per run. Next. So it's a robust PCR-based technology. It's qualitative, giving reliable results. It's relatively sensitive, with a 5% mutation allele frequency. The samples are very practical. It's a test that's linked to the study that proved the efficacy of the drug, so it identifies every potentially treatable patient, and it's very flexible in terms of laboratory throughput. Next. So it's actually looking at 13 different mutations, 1 mutation in exon 7, 7 mutations in exon 9, and 3 specific mutations in exon 10. Again, it's qualitative results. It's displayed in RotorGene Assay Manager software, informing the system operator if 1 or more of the 11 mutations detected are present. It's of interest now. We're identifying cases with more than 1 mutation, and the question is whether these respond better or not. I think additional data will be required. Some studies have shown as much as 15 to 30 per cases show 2 mutations. I'll just say in our hands, it's been about 2 to 5%, but it's a very interesting subset of cases. Next. And here's the intended use. So the full name is the TheraScreen PIK3CA RGQ PCR kit. Again, real-time qualitative PCR detecting 11 mutations in the PIK3CA gene using genomic DNA extracted from formalin-fixed paraffin-embedded breast tissue, or theoretically, at least, circulating tumor DNA from plasma-derived and EDTA anticoagulated peripheral blood tubes. They're processed using the FFP tissue kit by QIAGEN for manual sample preparation. For blood, there's a corresponding QIAGEN nucleic acid kit for manual sample preparation. And for both specimen types, it's the RotorGene Q RGQ MDX instrument used for the amplification with software detection. Next. And here's just a summary. You can use FFPE tissue, or at least in theory, plasma, although still not validated in our laboratory. You might understand how difficult it is going to be to validate the plasma in terms of sample procuration. Purification can be done either using the QIAGEN tissue kit or the QIAGEN circulating nucleic acid kit, depending on whether you use the paraffin or the blood. But the detection is the same on the RotorGene PCR kit on the RotorGene platform using the RotorGene TheraScreen PIK3CA assay software. And that's basically the process. Next. So to summarize, the TheraScreen PIK3CA RGQ PCR kit contains the reagent required to perform qualitative real-time PCR on DNA samples extracted from FFPE cancer tumor material, or theoretically from patient blood plasma. Here it says coming soon, and I think that's true. It reliably detects clinically actionable alterations in the PIK3CA gene, which are predictive of a response to the Pi3k inhibitor drug alpelisib. It enables sensitive and specific identification of actionable mutations, those 11 we enumerated, to identify patients suitable for treatment. We know that approximately 40% of patients with hormone receptor positive, HER2-negative breast cancer have PIK3CA mutations, and the Pi3k pathway activation is implicated in cancer progression and estrogen therapy resistance. And again, we have the PIK3CA CDx Mutation Analysis as an FDA-approved companion diagnostic intended to aid clinicians in identifying breast cancer patients with a PIK3CA mutation are eligible for treatment with alpelisib. Next.
Well, thank you, Dr. Weiss. Once again, you've given the attendees a lot of information. Let's see if they can provide us with a little bit more information on how they are likely to use the companion diagnostic. We'll open that poll and see what our attendees are responding to. You mentioned the NCCN guidelines. When were those updated, Dr. Weiss?
Well, one of my longstanding complaints is that the guidelines don't reflect clinical practice for a long time, that there's a long lag between when the guidelines come out and when the drug is approved and when the guidelines come out. But the NCCN has really been responding to that criticism, and they're getting really quick. I think within a month from the drug approval, it took for the NCCN to revise their guidelines. And that's just unbelievable. Unbelievably good, I might say. That's great. And the drug was approved in May, is that correct? I think it was May 24th or May 25th, around that time. Wow, so very fast action. Well, let's see what our attendees indicated. Compared to their previous practices, how will they use the NCCN recommendations how will they influence the DNA testing for breast cancer? So half of the respondents are more likely to order or perform DNA testing given those recommendations. And as you indicated, Dr. Weiss, this is sort of a moving target. It evolves over a period of time. Is this more or less what you would expect at this point? I think so. I think we're evolving towards the testing. It can't happen overnight. I think it's good that people need to get as much information as they think they need to make their own decisions. I think it's very important. And I hope they go out there and get that additional information. Super. Well, we're just about at the end of our presentation here. We'll have quite a bit of time for a question and answer period. We certainly want to acknowledge NeoGenomics. Let me ask and answer the first question. Yeah. The question I always get is what's going to happen when we get this blood assay up and running? Are we going to be doing that instead of the tissue? I think the tissue assay is going to remain the gold standard. I think if you look at the if you look at the validations by QIAGEN, the assay in plasma misses a substantial subset of the cases. So if you were to do the plasma and you got a positive result, I think that would be very helpful. But if you were to do the assay and got a negative result, and you're going to expect a negative result in most cases, first of all, 60% of the cases are negative anyway. And then a substantial subset of those 40% that might really be positive are going to be negative in the plasma testing. You're going to find that you're going to be reflexing from the plasma testing in a large majority of cases. So I'm a tissue guy, but I think there are cogent reasons for preferring the tissue test. It's going to remain the gold standard. I think people may become frustrated with the plasma test because the negative result, which you're going to obtain, in the large majority of cases, is going to necessitate reflex testing to the tissue anyway. When would you do the plasma test? When the tissue is not available. But in breast cancer, it's not like lung cancer. The tissue is usually going to be available. So I get asked, can you use archived specimens? Can you use the original breast cancer to do the testing, even though if it was obtained 2 years before? And the answer is yes. The mutation is relatively stable. I won't say there are not exceptions where mutations can develop that were not present in the original tissue, but for the vast majority of cases, that mutation is stable. And if you don't have a recent metastasis, I think it's okay to go back to the original tumor tissue.
You mentioned that the plasma is not validated in your lab yet. What do you think is the pace of that, given that, as you suggest, that tissue is likely to remain the primary specimen type for most diagnoses?
We'll have it in a few months. It's hard to get our hands on, plasma with PIK3CA positive results, for the independent validation. We'll have it, but even after we have it, again, tissue is going to remain the gold standard. And the only real reason for going to plasma is when you maybe have only bone metastases and you can't get a specimen because it has to be decalcified, or somehow that tissue is not available, the patient can't be biopsied and you can't get the previous specimen for some reason. But I think even after we have the test, we're going to strongly recommend tissue as the gold standard.
Mm-hmm. One of our attendees asks, do PIK3CA mutations ever occur in HER2 positive breast cancers? And if so, is there any information about treatment data in that case?
I don't know the answer to that question. I would assume that you can see PIK3CA mutations in that population, but I don't believe it's going to be as high as in the HR-HER2 negative population. And right now, there's no treatment implicates that because alpelisib is only approved in cases that are hormone receptor positive and HER2 negative.
Mm-hmm. You mentioned that if a patient's testing was done some time ago, that those mutations would still be valid. So am I understanding then, if there's been true tumor profiling performed in the past, that there's no need to retest with a more recent sample?
Certainly, if there was a PIK3CA mutation identified previously, there is absolutely no need for retesting. But even with a negative result, it should be pretty reliable.
Mm-hmm. I'm looking for additional questions from our attendees. who are not forthcoming with a lot, which I think that speaks well for the clarity of the presentation, but we certainly want to answer any questions that the attendees have.
Yeah, I'll take that as a compliment.
Absolutely. I wondered about the PI3K pathway. Is it exclusive to breast cancers?
No. PIK3CA mutations are one of the more common mutations in any tumor. It has implications in colon carcinoma and a lot of other solid tumors. It's got to be about the second or third most common mutation that we see in the actionable mutation that we see in solid tumors in general.
Mm-hmm. Does that mean we're likely to see an expansion of these indications into other types of cancer? And will the same companion diagnostics be useful in those settings? Clearly, they would have to be retested and validated, and lots of studies go on. But what's your sense of that future?
Yeah, it's complete speculation, but I would see an expanded role in the future. Mm-hmm. I wouldn't be surprised if pharmaceutical companies are working on this right now.
Yes. One of our attendees asked, are these mutations present in ductal and lobular types of carcinoma?
Yeah, so it's particularly common in hormone receptor positive, HER2 negative cases. Those are the huge majority of both lobular and low-grade ductal carcinomas. So if they're advanced, if they've recurred or metastasized, that's the perfect population to be studied.
Mm-hmm. We have another question that I can answer. One of our attendees asked, will the slide deck be made available? And the answer to that is yes. A PDF version of the slide deck will be available to attendees, and you can find that in a couple of days when the presentation is archived, and it will be attached to the handouts section. And I wanted to mention also, I think I neglected to mention earlier, if you're interested in Dr. Weiss's background, I encourage you to read his bio, which is also in the handouts. And he's had quite an illustrious career and continues to make waves. So thanks for that. But check out Dr. Weiss's bio in the handouts section of the chat box. I'm looking here for additional questions, and I don't see any. Again, I think that speaks to the clarity of the presentation. So I think we're about done for the day, and we can move on and close out and let everybody go have their lunch on the West Coast, because it's certainly lunchtime. But we do want to thank our sponsors, QIAGEN, for supporting the presentation today. And in case you were in any doubt, of course, the views expressed herein are those of the speaker and don't necessarily express the views of QIAGEN. And there are some additional disclaimers here that are important for our attendees to understand. Certainly, for more information about the companion diagnostic, contact your QIAGEN sales representative. They can tell you all of the situations about that product. And here is some contact information for Dr. Weiss, which I encourage you to use if you have questions specific for him. God forbid you should have to contact me, but my contact information is there as well. Certainly, there are a number of potential webcasts out there in the air, and we would love to help other companies do their presentations, so feel free to contact me for that. In a couple of days, the webcast will be available on demand. You can revisit it by logging in via the CLP website, or you can go to the registration page using this short link. So that pretty much does it for today. I'm going to read progress past this slide just to show our attendees that at the end of the presentation deck are the references for all of the slides today, and that information is also one of the handouts. You can find all of the references in the handout section, but here is at the end of the deck here are all of the references for today's presentation. So one way or the other, you'll be able to track down all of these sources of information, and that again speaks well to the depth of this presentation, that we've got so much backup on it. So I think that will do it for today. We've just run out of time, and we certainly want to thank all of our attendees, and we will see you again at the next presentation.
Thank you very much.
Thank you, Dr. Wise. Okay, well, I think we're done.
I'm still here.
Good. Yeah, we're closed out, and good presentation.
A little disappointed on the attendance, but it's a specialized diagnostic.
Yeah,
so,
I'm encouraged that we had so many people register. It probably does mean that they will come back and look at the archived version.
Hopefully, very good.
Yeah.
Are you satisfied with it?
Yeah. All right. Well, thank you very much. I don't want to take up more of your day. We'll circle back with Nicole and with the folks at QIAGEN to make sure that they know that the archived version is available, and you'll get that information as well.
All right. Terrific. Thank you very much.
Thank you. Bye-bye.
All right, bye.